So good morning, everyone. And my name is Prashant Daur. And today I'm going to talk about the peptide therapeutics analysis by capillary electrophoresis. So this is the overview of my presentation. So I'm going to start with the principle of capillary electrophoresis. And in this section, we will talk about how capillary electrophoresis works and what is the principle behind of the capillary electrophoresis. After that, we will discuss about the features of capillary electrophoresis, and then we will talk about different applications of CE in peptide analysis. And in the last, we will also talk about a new technology, which is called CSI mass spec for the peptide analysis. So in this section, we will briefly talk about how CE can be connected with the mass spec for further peptide analysis. So before starting the presentation, we will talk about what is CE and how it works. So capillary electrophoresis is basically a electrophoresis technique. And electrophoresis is a separation technology which basically separates the molecule based on the charge on the molecule. So since we are doing electrophoresis with the help of capillaries, so we call this capillary electrophoresis. So when we talk about the separation in CE, we always consider the electrophoretic mobility, which is the mobility of any charged molecules when we apply the voltage. So what happened here is, if you see, we have two different kind of electrodes in capillary electrophoresis. One is anode and second is cathode. And in this figure, you can see we have 5 different kind of molecules, and we are considering the size is same. Only difference between these 5 molecules are the charge. So if you see in the red circle, they are positively charged. One is double charge, and then second one is single charge. The white one is the neutral molecule, and then last two one is single negative and then double negative charge. So that means we have 5 different kind of molecule in this example, which has different kind of the charge. So what happened when we ran this kind of molecule in capillary electrophoresis, we need to apply the voltage to get the separation. And when we apply the voltage, the highly charged molecule will travel fastly to the respective electrodes. So for example, if we apply the voltage, the 2 positive molecule will move faster towards the cathode, and 2 negative molecule, which is highly negative, is moving towards the anode. So if you want to calculate the mobility of any molecule or any ion, so we have this formula which basically talk about the electrophoretic mobility, which is directly proportional to the charge of the molecule and inversely proportional to the viscosity of the solution and stoke radius of the ions. So that means if you increase the charge, the mobility will increase, or if you decrease the charge, your mobility will decrease. Furthermore, if you increase the viscosity of the solution, that means the buffer or the sample, that means your mobility will decrease. So here we have the features of the capillary electrophoresis technology. So the first one is the capillary electrophoresis will give you exceptional resolving power. So why we get the exceptionally resolving power? Because the capillary, what we use for CE applications, is made of bare fused silica, and it has very, very less diameter. So we are talking about the diameter of the capillary, which is in a micron. So we have 25 to 100 micron capillary. Because of this small diameter of the capillary, we always get exceptional resolving power. So as we discussed, these capillaries are very small in diameter. So because of this, we will getting more than 1 million theoretical plates when we run any sample in capillary electrophoresis technique. So if you want to compare the CE with HPLC, just wanted to give an example. In HPLC, the number of theoretical plates is somewhere around 10,000, but in CE, it is more than 1 million. So because of this high number of theoretical plates, we always get a good peak efficiencies and exceptional resolving power. The second most important feature is the requirement of the samples for the sample injections. Since the diameter of the capillary is very small, we need some nanoliter of the sample for any kind of CE experiments. And the next feature is minimum wastage requirement, because what happens is the diameter of capillary is very small, and we inject very small amount of buffer to get the separation in CE applications. So just take an example of any of the experiment. So 1.5 to 2 ml of the buffer is sufficient to run around a couple of samples, like 50 or 60 samples. So that means you have a green technology where the wastage is very minimum. We do have so many applications where you can use the capillary electrophoresis instrument for the DNA and protein analysis. Just take an example of the gene therapy applications. So for DNA and protein analysis, we have dedicated instruments, hardware systems, where it will deliver the gel to separate the DNA and protein molecules. So CES is always considered as a complementary technology to HPLC if you're working in HPLC for the peptide analysis. And if you're getting any challenges on the separation of any kind of peptide impurity, so always you can consider capillary electrophoresis as an orthogonal technology. So this slide schematic shows the view of a P800 plus pharmaceutical instruments. So basically, this is the capillary electrophoresis instrument. And on the top left, you can see the cartridge, which basically contain the capillary for the separation. And the length and the diameter of the capillary can be changed or adjusted based on the molecule complexity or the resolution what you are looking for it. So then we have a software which is called a 32-karat software. And this software is the one and only the software to calculate, to quantify a molecule and to run the software to the instrument. So we have a dedicated software which is a 21 CFR compliance, and this can be used in the QC labs or GMP environment. To get the reproducible data in capillary electrophoresis, we have a technology that is called the liquid capillary coolant. This technology will give you a reproducible data, and this is a patented technology from the SCIEX. So what happened is, since you are applying voltage in capillary electrophoresis instruments to get the separation, so that will convert into the current and that will generate the heat. To get the reproducible data, this capillary coolant will depreciate the heat and it will maintain the reproducibility throughout the run. On the right-hand side, you can see we have a modular detectors. This will give you a more flexibility in terms of the choice of detector you want to run for the different applications. So we have a UV detector, we have photodiode array, and then we have third detector is laser-induced fluorescence. So laser-induced fluorescence detector normally we use for a molecule kind of example like glycan or sugars, where you can tag your molecule with some fluorescent dye and then you can detect on laser detector. We do have a temperature control capacity for the sample stability. So if your sample is very fragile or there is any stability issue for the sample, you can store the sample at 4 degrees centigrade inside the instruments. So for high throughput sample analysis, we have 2 buffer trays. We call this inlet buffer tray and outlet buffer tray, where you can keep all those buffer required to run any application in CE technology, and you can get high throughput data analysis. As we discussed in the last slide, you can also analyze DNA and protein applications in capillary electrophoresis. So for this application, we need a high pressure pump to deliver the DNA and RNA gels inside the capillary. So we have a pressure pump inside the system. So when we talk about the different separation modes in capillary electrophoresis, we have different modes in capillary electrophoresis for the peptide analysis. The most common are capillary zone electrophoresis, which is the capillary zone electrophoresis is a method to quantify and identification of the charge variance of your peptide molecules. And this can be used for purity analysis, quantification of charge variance analysis in any peptide samples. The most popular method, the second method, is capillary isoelectric focusing, where we can calculate the PI of your charge variance in the peptide, the molecule. And this method is very good because this will give you ultra high resolution data in terms of PI calculation. The next application is capillary isoelectric electrophoresis. This method is basically we use when we connect CE with the mass spectrometry. And this is one of the method which is good for the pre-concentration of the sample. So if your sample has any challenge with the concentration, if your sample is very, very less concentration, so inline or inside the capillary, you can concentrate the sample in CITP application, and you can run with the mass on the mass spec. The fourth application or the separation mode is MEKC, which is called micellar electrokinetic chromatography. And the principle of this MEKC is based on hydrophobicity of the molecule. And basically, if you have a challenging molecule which doesn't have any charge, like neutral molecules, so the MEKC is a good method to separate those kind of molecules. And the principle of this molecule is based on the differential partitioning between micelles, which is normally created by using a surfactant like example like SDS or HPMC. The next application is capillary SDS. So this application is very good method to distinguish or to establish the differentiate to characterize the molecule based on your size heterogeneity. So you can calculate the size of your peptide or the impurities, and you can also check the purity of your main peak, and you can also calculate the size of your molecule. The limit for this method is 10 to 225 kilodalton. So if your peptide falls in between this range, so you can use CESDS application for size-based analysis. So when we talk about the applications of CE in peptide analysis, so we have so many applications where CE can be used for the peptide applications. So the first one is the peptide mapping. Yes, you can use CE for the peptide mapping. The next application is peptide identification. So here you can separate different impurities level present in your peptide, and you can identify those peptides. You can also check the purity of the peptides if you're working in a QC lab or a GMP lab, if you want to quantify the impurity of the peptide. So the CE is a great tool. So when we are doing or when you're working on the peptides, so we always talk about the stability of the products. So if you want to understand the degradation behavior of any of the peptides, so you can use the capillary electrophoresis technology for the degradation studies. You can also use CE for stereoisomer analysis, and this basically is one of the great tools to characterize the chiral analysis of the peptide molecule. Then we have other applications like you can use CE for the bioanalysis of physiological peptides. You can also determine the reaction kinetics, and you can also determine the pK of your peptide. So I will talk about, in this channel, I'm going to talk about the charge variant analysis of the peptide. So here we have two different methods. One is capillary zone electrophoresis, and another one is capillary isoelectric focusing. So we'll start with the capillary zone electrophoresis. So capillary zone electrophoresis is a very fast method. We call this rapid capillary zone electrophoresis. And here the separation is based on charge to hydrodynamic radius of the peptide molecule. So what you can see in this figure is we have two different electrodes, anode, which is positive charge, and cathode, which is negative charge. So if you see in the picture, we have different color-coded charged molecules. And when we apply the voltage in the capillary, this molecule will start moving towards the cathode, because the highly positive and the less size molecule will start migrating first in the capillary, and then you will get the separation in capillary zone electrophoresis technique. The capillary zone electrophoresis technology is a very good method. It will give you high resolution method for the charge variance analysis. So basically, you can distinguish between basic and acidic variance, which is present in your peptide samples. You can also do the native state analysis, and this capillary zone electrophoresis we have for this application, we have a dedicated case. That means the method development is very little. So you don't need to develop the method for the CZD analysis, because the kit contain all the required buffers in the capillary to run the CZD application for the peptide. The sample preparation is very easy. No desalting step is required. Just dilute the sample, make it 1 mg per ml, and then you can directly shoot into the capillary electrophoresis instrument. The data quality, what we get here, and the throughput, what you can achieve is very good when we compare with the IEX technology. So the capillary zone electrophoresis is again a very good technology for the charge variant analysis. So I have a couple of good example data where we ran some of the peptide molecules to quantify and to check the purity of the product. The first one is insulin. And so in this experiment, we have taken 0.6 mg per ml of the insulin, and then we ran this molecule on the capillary electrophoresis method. So what you can see here in this picture, that the separation of the main compound, where you can see the migration time of this main compound is 2.55 minutes. And then we can also see one of the impurity which is separated at 2.67 minutes, which is A21D amino insulin. So this is the impurity of this particular degradation. You can call this a degradation product of the insulin, which is well separated with the main compound. You can also see on the left-hand side the emprisol, which comes from the formulation buffer. So the take-home message from this slide is the runtime. If you see the runtime is very short. Within 4 minutes, you will be able to achieve the separation of the charge variance of the insulin molecule. So the next example is the quantification of somatropin. Somatropin is growth hormone, and here you can see the charge variance distribution of two different somatropin molecule. And these are the standards which got from the USP pharmacopoeia, and the second one is from the European pharmacopoeia. The red trace is from the USP pharmacopoeia, and then the blue one is from the European pharmacopoeia. So what we have done here is we compared both the standards by using the capillary electrophoresis. And for your information, the somatropin is a 191 amino acid protein, and the size is 22 kilodalton. So what we achieved here is we got a separation of different charge variants, which is labeled as P1, P2, P3, P4, and in case of USP, you can also see the P5. So in the center, you can see the main peak, and on the left-hand side, you can see the basic charge variance, and on the right-hand side, you can see the acidic variance. So if you look into the data again here, the P2 stands for the cleaved form, which is a cleaved form of this hormone. And the P3 is a mutated, is a mutant form. And the P4 is a deamidated form, which is a key impurity to monitor in these kind of applications. So what we got here is P1, P2, P3, and P4, different kind of charge variants for this hormone. So the distribution of charge variance between the two standards are slightly different, because in the USP we got one extra impurity, which is P5, but the migration time or the relative migration time of all the impurities are the same and fall in the range specified by the European Pharmacopoeia, which indicates the identification of the impurities, which is very consistent throughout the two different standards. So now we will discuss capillary isoelectric focusing, and this method is very good method to quantify the PI value of the peptides. So in capillary isoelectric focusing, the separation is based on isoelectric point. And here what we do is we take a neutral capillary, and then we take the sample or the peptide sample. Then we make a master mix of ampholytes. This ampholytes is basically a mixture of acidic and basic species. And then we add ampholytes, protein or peptide, then a few stabilizers, and then we inject inside the capillary. And then we apply the voltage. So when we apply the voltage, the pH gradient will form inside the capillary, and the separation will happen based on isoelectric point. So for this application, we use phosphoric acid as analyte and sodium hydroxide as catholyte. So this is one of the classical example of PI determination of exenatide. I think this molecule does not need any introductions. This is very famous molecule, and exenatide is a GLP-1 receptor. It contains 39 amino acids, and the size you can see here, it is close to 4,000 kDa. So what we have done is we took the exenatide standards, and we ran with the CIF method. We also spike five different PI markers to calculate the exenatide PI value. And in the left-hand side, you can see the PI markers, which is denoted as 10.1, then 9.4, 7, and 4.1. In between, we have this peptide the exenatide, where you can see the PI value of this molecule, which is coming around 5.6. So this way, you can calculate the PI of any of the peptides by using the peptide PI markers. And this method is also very reproducible. This will give you ultra high resolution data. The second example, what I have is the separation of the glatiramer acetate. And what we have done is we compare the different batches of this glatiramer acetate by using the capillary isolated focusing. And you can see here the separation of different batches of glatiramer acetate. And basically, this kind of data will give you idea that this kind of molecule contain the polydispersity, and you can see the changes in polydispersity in glatiramer acetate. You can also calculate the PI value of each and every peak by using the peptide markers, and you need to spike the peptide markers. These peptide markers are the known PI values, which comes with the CIF kit, and then we can calculate the PI values of each and every peak. This method is also a very good method for the QC and the GMP laboratory practices. So this can be used for the product release also. So this is again a very classical example of the liraglutide, and we have taken here the Victoza, which is a reference standard. And what we have done here is, again, we wanted to check whether this liraglutide contain any kind of charge variants. So we ran the molecule by using the capillary isoelectric focusing, using the ampholytes, and then what we found here is we can see and we can able to separate different charge variants present in the liraglutide. So on the left hand side, you can see the basic species coming at around 41.95 at migration time. You can also see the PI value, which is there. And for the first peak, it is 5.1. And then we got two M-shaped main peak and small impurities you can also see on the right-hand side. Basically, these are the acidic variants which is present in the sample. So now we'll talk about the ion analysis by using the capillary electrophoresis technology. So ion analysis by capillary electrophoresis is a very important technology to quantify the anions and cations present in your formulation buffer or the sample. So why we need to calculate or to quantify the ion analysis by CE or any other method? Because the regulatory agencies like FDA and European Pharmacopoeia require that pharmaceutical products should be tested for composition of their integrity, their identity, strength, quality, and purity of the active as well as for the inactive ingredients. So we want to quantify whatever ions presents in the formulation buffer. So this is the FDA and European Pharmacopoeia requirement. And the basic drug may also contain inorganic salts or organic acids, so that can also be quantified by using the capillary electrophoresis methods. So some acidic drugs also contain the methyl cations, so that can also be analyzed by capillary electrophoresis. So for anion and cation analysis, we have two different dedicated methods where you can separate the anions and cations which is present in your sample, any peptide samples of formulation buffer. So this is an example of analysis of organic and inorganic anions. And in the first electrophoregram on the top, you can see the separation of organic anions. You can see what we have got here is different anions like chloride, then azide, formate, succinate, and then valerate. So this analysis was done at UV 230 nanometer. And the best part of this method is you can go up to ppm or ppb level, and the runtime is less than 8 minutes for this ion analysis. The second example on the second electrophoregram, on the bottom, you can see the separation of inorganic anions. And here we were able to achieve 6 different standards of inorganic ion. That means what we got here is chloride, nitrate, sulfate, azide, fluoride, and phosphate. So these are just few of the example, but you can separate any of the anions which is present in the sample by using these methods. For the analysis of the cations, we use UV 200 nanometer, and in this electroprogram, you can see the separation of the standards. So here we got ammonium, sodium, potassium, lithium, magnesium, and calcium. The runtime is again a very short, and less than 5 minutes, we were able to quantify and identify of cations which is present in the sample. So if you talk about the comparison between two technology, which is capillary electrophoresis and ion chromatography, so we have the data here. So we compile in this table. And so in the left-hand side, you can see the benefits of both the technology. And then in the center, we have capillary electrophoresis, and then in the right, we have ion chromatography. We do have a different electrophoregram and chromatogram to show you the difference between the resolution of both the technology. So first, we will talk about the different benefits. So when we talk about the risk of the technology, so capillary is basically is open tube. So whatever you inject, it will come outside from other side. So that means if your sample is dirty, or if your sample contain a lot of salt, so it will not damage your capillary. So that means the sample preparations is very easy in case of capillary electrophoresis analysis. When we compare with ion chromatography, so sometimes there is a challenge, and sometimes the column will be damaged because if your sample contains a lot of salts or the matrix is not clear. So when we talk about the capillary electrophoresis is a very fast method, and less than 4 minutes you can achieve, or you can separate all those anions and cations present in the sample. In other hand, for ion chromatography, separation takes around 12 to 30 minutes. Again, depends on the column, and and for analysis in anions and cations, sometimes you need different kind of columns, and each and every column require the equilibration. But in case of capillary electrophoresis, there is no conditioning is required for a sequence run. You can just clean the capillary with some kind of buffers, which is present in the kit, and then you can start the analysis. So when we talk about the flexibility, the capillary electrophoresis is one instrument, but it can be used for the different applications, like what we discussed. Like you can use for chiral analysis, you can analyze for the purity, for the charge variance analysis, and of course for the anion and cation analysis. We do have many applications where you can use for other regular molecules like monoclonal antibody, and you can also analyze the gene therapy, the products. For ion chromatography, it's a dedicated column for each application. As I said, for different chemistry, you need a different column in ion chromatography. So the capillary electrophoresis is a green technology, use only few ml of non-toxic buffers for the large number of samples. And for ion chromatography, you need so many different kind of buffers, and the volume is always is smaller than what we consume in sea. So it's a green technology. When we talk about the separation in both the technology, in the electrophoregram on the right side, you can see the first figure is from the capillary electrophoresis, where you can see the baseline resolution of different ions. And this was the example of the cation. We were able to achieve the resolution of all the standards which is present in the sample, and the runtime is almost less than 4 minutes. So when we talk about the ion chromatography, the resolution is not good, the peaks are not well separated, and the runtime is more. So now I will talk about the different application, which is the chiral analysis. As you know, the antibody analysis has some challenge because these are the stereoisomers, and which contain different asymmetric centers, and these are basically they form the mirror images. The chirals are physically identical, but optically they are different. So whenever we talk about the enantiomer analysis, there always be a challenge because of the behavior of this molecule. For chiral analysis, we use cyclodextrins, and there is two different kind of cyclodextrins present in the market. The first one is static cyclodextrins, and then second one is charged cyclodextrins. So for CE application for the chiral analysis, we use charge-based cyclodextrins. These are the dynamic cyclodextrins that moves inside the capillary, and they are highly sulfated. So we have three different kind of cyclodextrins presents in the market. One is alpha, beta, and gamma. And alpha, beta, and gamma all contain different kind of sulfate group in their core. These cyclodextrins are negatively charged, and this will increase the resolution when you analyze the chiral molecule by using the cyclodextrins. So here I have an example of the chiral analysis by using capillary electrophoresis. So here we have taken the glyceride as an example. And in this lecture program, we can see the method development activity then for this molecule. So what we have done is we have taken a gamma cyclodextrin for this experiment, and we prepared different concentration of cyclodextrin starting from 0 to 20 millimolar. And you can see the separation of D and L form of this molecule. So in the first example, which is generated in the blue, where we have a 0 cyclodextrin, so we were not able to separate these two chiral compound, but when we increased to 20 millimolar cyclodextrin, so we were able to achieve the separation of D and L isomers. So this way we can optimize and develop the method for any challenging peptide molecule, and then we can separate the different isomers. So for when we talk about the LOD for this method, so for this molecule, we got 0.02%. So another is classical example of two different molecules for the chiral analysis, and here we have taken ketoprofen, labetalol, and left hand side you can see the separation of ketoprofen, and in the insert you can see the separation done by the HPLC. But if you see the resolution what we get here in C, it is much more better than HPLC for the both example. On the right hand side in the electroprogram, you can see the HPLC separation and the CE separation. And here also you can see the resolution what we got for the different peaks which is present in this sample. So again, if we compare the CE with HPLC for the chiral analysis, so we have several advantages over HPLC. So the first one is, again, the capillary can be used for any challenging samples. So no need for desalting or to remove the matrix. In case of HPLC, it can damage your column. So you need to take care of the sample preparation. Flexibility, you can optimize the capillary. There is 3 buffers required for this analysis, multiple columns required for HPLCs. When we talk about the method developments, so these methods are already optimized. So we have a methods which contain the different steps, even though this will also contain the buffer required for the analysis. So you don't need to adjust the pH. You don't need to identify the different components which is required for the analysis. But in case of HPLC, the method development is required. And this is the last point what we already discussed, since it is a green technology. So it's in CE, only millimeters of the sample we required, milliliters of samples of required for the analysis. So there is no wastage of the buffers. So in this section, I would like to talk about the capillary isoelectric focusing technology and capillary zone electrophoresis technology, which can be alternative for the different applications. So that means the CE can be connected with the mass spectrometry to get the information what we are getting from the standalone technology, and this can be used as orthogonal to LCMS. So what we do here is we combine the capillary electrophoresis with ESI nanosource and with the mass spectrometry. In this example, you can see the capillary electrophoresis on the left-hand side is coupled with ESI nanosource with the mass spectrometry on the right-hand side. So C is ultra low flow rate, separation technology, which is coupled with the high resolution or high sensitivity mass spectrometry, and 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 then you can when you connect CE with ES with any of the mass spectrometry, so basically because of ultra low flow rates, which is less than 30 nanoliter per minute, you will achieve a greater resolution and the sensitivity will also improve. So this is a schematic of the CSI cartridge. We call this CSI Optimus cartridge. And from this, you can connect the CSI or the C instrument with the mass spectrometry. So we have a dedicated instrument which can be used as the standalone CE for different applications for the peptides, or it can be coupled with the mass spec as a front end. So this model is called CSI 1000, and this figure you can see we have two buffer plates on the left hand side and right hand side where you can keep the buffers. Then we have two different capillaries, and the longer capillary is a separation capillary, and then shorter capillary is conductive capillary. We do have a high voltage power supply to get the separation for your peptide molecule in case of CESI and the mass spec experiment. And this CESI Optimus cartridge has a spare housing from where you can get the spray and which will connect with the mass spectrometry for the detection. So CESI is an integration of capillary electrophoresis with electrospray ionization into a single dynamic process within the same device. So in this picture, you can understand more about how the CSI technology works. So basically, we have a capillary which is made of bare fused silica, and then what we have done with this bare fused silica capillary is we etch the capillary ends with hydrofluoric acid to become it more porous. So when we inject the sample, it will give an ultra low flow of your sample and the buffer. And then the second capillary, which is the shorter capillary in the previous picture, we fill the capillary with the conductive buffer. Just take an example of the conductive buffer. What we use for the CSI experiments are like acetic acid of the different concentration. So when you inject the sample inside the capillary, in the separation capillary, we apply the voltage in both the ends of the capillary, and then your Optimus cartridge will deliver the ions, and these ions will go inside the mass spectrometry for the separations. So we apply the voltage from the ESI source to form the spray, and then for the separation we apply the voltage from the CE instruments to get the separation of ions based on the charge and the size of the molecule. So this technology will eliminate the interference from oxidation and the reduction, and it will enhance the sensitivity of the separation. So basically, it increases ionization, and it will also decrease the ion separation, because the flow rate what we are talking about is in nanoliter per minute. So when we talk about, or if you want to see the influence of low flow rate on the sensitivity, so here we have one example. In this example, we injected angiotensin at 2 nanogram per ml in 10% acetic acid by using the CESI and the mass spec. And what we have seen here is when the flow rate is very high, which is, you can see on the right hand side, which is around 350 nanoliter per minute, which is called the high flow, the sensitivity is very less. And then when we are going towards the low flow rate, which is 10 nanoliter per minute, which is ultra low flow rate, which can achieved by using the CESI technology, the sensitivity is significantly improved. So that means when we decrease the flow rate, when we are using a very low flow rate, the sensitivity is increased. And this is a main advantage of using CESI connection with the mass spec for peptide analysis. So I have a few examples for the CSI-MS analysis, and we will talk about the first example, which is neuropeptides such as VIP and PCAP analysis done. This analysis was done by using CSI and mass technology. So normally these kind of challenging peptides traditionally run by radioimmunoassay and or immunoassay. So this technology we call traditional technology has benefits like the method is very sensitive, and it also it also has a drawbacks like you need a multiplexing technology to differentiate if you have a different kind of peptides presence in the same sample, or sometimes it is very difficult to optimize the method when you are running in immunoassay or radioimmunoassay. So sometimes specificity can also be a problem, or it can be challenging when we talk about immunoassay or radioimmunoassay for peptide analysis. So what are VIP and PCAP, the peptides? So VIP is called vasoactive intestinal peptide. It's a very basic peptide hormone, and it's a neuropeptide containing 28 amino acids residue. The molecular weight you can see on the right hand side for VIP. The PCAP peptide is pituitary adrenalized cyclase activating polypeptide, also known as PCAP, is a neuropeptide which contains 38 amino acids, and the structure is similar to VIP. You can also see the size of this PCAP uh, peptide in this screen. So what you can see here is 28 amino acids for the VIP and 38 amino acids for the PCAP. This color coding blue and red basically demonstrate the blue one is the basic amino acids and the red one is the acidic amino acids. So sometimes what happens is if your peptide contain high level of basic amino acids, so this kind of molecule makes difficult to analyze by conventional LCMS technology. So if your molecule or if your peptide contain basic species, so sometimes you need a good method development and challenging approach in terms of LCMS approach. So here you can see the separation of VIP and PCAP peptide by using the CESI. And you can see the baseline resolution of both the peptide. On the left hand side, you can see the VIP, we got a separation. And on the right side, you can see the PCAP molecule, which is also separated by using the CSI method. For this method, we used base fused silica capillary, and sample was injected in a reverse polarity. So we use around 2 psi for the sample injection, and the separation was done by using 30 kV voltage. We also done the calibration studies for both the peptide. So here you can see the example of PCAP, the molecule. So calibration range covers detection limit, and for this molecule, whatever the detection limit required, we achieved. And here in this electrophoregram, you can see we have ran the sample. We have ran the calibration studies using 1 to 200 nanogram per ml, which is close to 440 femtomole to 40 picofemtomole per ml of the sample. So we got a very good calibration studies here, and we also calculate the R square to check the linearity, and we got 0.999 in this case. So this is the results from the analysis of low-level standards. So we spiked the different concentration of standards to check the LOD or the sensitivity of the instruments. We prepared the sample by using phenol acetic acid and 100 millimolar acetate. So for the PCAP, we ran 3 different runs. One is blank. The first one is a blank. Second is 440 femtomole per ml, and then third one is 2.2 full per ml. For the same studies we have done for the VIP, for the same 3 different kind of concentration we took. And if you see for the PCAP on the blank, we didn't get any peak as expected. And at 440 femtomole, we got a separation of PCAP, and you can see the red circle, which basically tells about the signal to noise ratio. For the VIP also, the blank, there is no profile, and we also achieved 440 femtomole for the VIP. So for this application, we injected around 20 nanoliters of the sample for analysis of the different concentration of PCAP and EIP samples. We also done the studies by using the serum sample, and here you can see the workflow for the serum samples. So we started with the SP column, and we took around 1 ml of so we have taken 6 ml of the sample, which is 200 milligram, the serum sample. Then we have done the washing steps with different buffers, and the last we got a sample which is around 200 microliter, and this serum sample was analyzed on the CSI-MS for further analysis. So this is a workflow. It's a very simple workflow, and it takes very less time to analyze the serum samples. So what we achieved here is, if you see here, we have two different kind of PCAP sample. One is denoted as channel 1, and second is for the channel 2, where basically we just wanted to confirm the studies what we are doing it. So we ran two different sets of samples, and again in the blank you can see we got a very small peak, and then 200 femtomole we were able to achieve the separation of PCAP with a very good signal to noise ratio. And for the second sample set, you can also see there is some peaks which is present in the blank, and then 200 femtomole also we got a very good separation and with a good signal to noise ratio. So the peak the peaks which can you see in the blank, which are basically the contaminated forms of the serum. So in the next run, what we have done is we increase the rinsing steps in the capillary to avoid this kind of carryover, and we were able to achieve a very good profile by enhancing the rinsing steps. So this is I just wanted to show you this example because the method development activity is also possible in case of CSI-MS. And if you have a challenging sample like serum or the plasma samples, so you can optimize the methods. So these additional peaks are basically due to the serum, and it may have caused problem in radio immunoassay technology. But in case of CE, if you enhance the rinsing steps, so basically you will not get this kind of profile in the challenging samples. So if you talk about the comparison between CESI with LCMS, so we have done a study on the insulin, and here you can see the separation of insulin by using the CESI-MS and LCMS. In the LCMS experiment, the insulin you can see elutes at around 6.8 minutes, and you can also see earlier peak, which is eluting as a sharp peak, which are basically the solvent peaks. And on the same electrophoregram for the LCMS, you can see some peaks are coming on the last, which are basically high organics and of the gradients. So when you see the CSI profile, it's a clean profile, and here the separation is based on charge and size. So what we get here is the insulin peak, which is a major peak coming around 9.44. And then you can also see the various small peaks. We will talk later about these small peaks, but this is a degradation products of this insulin molecule. To check the degradation products for insulin, we done the extended experiment. And excuse me. And the first example, you can see the CESI-MS separation. And the second example, you can see the separation by the LCMS. So the region around the parental peak for insulin has been expanded to show you the resolution of the major impurity peak, which was formed as a result of deamidation modification. And in CSI-MS, this peak migrates before the parent peak, and for LCMS, this peak elevates after the parent peak. In both cases, the demyelination resulted in a small mass change of 1 AMU, but also resulted in a change alterations. So if you see the LCMS profile, and if you compare with the CSI, the CSI-MS gave better resolution than LCMS with the basal separation of this product from the parent molecule. And the separation of the major product is also very important. And when we talk about the small impurities like this degradation species, it is well resolved in CSI-MS. So in this profile, you can see the expanded region containing cleaved product from breakage of the insulin backbone. In ESIMS, in CSIMS, it shows the region where products from the breakage of the insulin backbone, which elutes and migrates. And in the CSI, the major peak was eluted around 7.2. The degradation molecule is separated around 7.2 minutes. We can also see the small impurities which is present in the sample by using both the methods. In LCMS profile, you can see small peaks which is coming from the gradients or the buffer. And in conclusion for these experiments, we can conclude that the LCMS and CSMS can be shown the complementary techniques because of their differentiating mechanism of the separation. Both technology were capable of detecting low-level degradation products, which were less than 0.5% of the area of the insulin peak. So what we achieved here is the impurity what the sample contain is less than 0.5%. And this study CSIMS exhibited better peak resolution for the degradation products of insulin and was capable of detecting additional features which was not detected by the LCMS method. So basically, you will be able to achieve, you will be able to get more resolved peaks for the degradation studies done for this insulin. So the next example is the analysis of epitopill. This is again a peptide. And you can see this, for this study, we have used the same CSI technology, which is also called the sheetless CESI-MS. And the main peak, if you see here, which is coming around 13.5 minutes in the first figure, which was further analyzed by INSO's CID-MS experiment. And we can here see the different sequence, which is confirmed by the mass spec on the MSMS level. And in addition to the main peak, the main peak of this peptide, we can also able to separate 15 impurities of this peptide, which is you can see on the right hand side table. So these are the different peptide impurities which was present in the sample, and we were able to achieve the separation of these small impurities by using the CSI-MS technology. So the best part of this method is the impurity what we're talking about, the 15 impurities which is present in this table. The concentration, the area of what we got for this is more than 0.05%. So that means the peptide which is coming here, if you see the peak area, it is more than 0.05%. So this peptide we calculated, this 0.05% comes from the pharmacopoeia, because as per the pharmacopoeia, when we talk about the peptide impurities, if any of the impurities presents more than 0.05% in the sample, then we need to characterize and identify those peptide impurities. So here you can see a direct comparison between nano RPHPLC versus CESI-MS. And in this example, we ran both the samples by using both the technology. So as a starting point, we take the peptide sample as a concentration of 25 microgram per ml, and total injected amount for nano LC was 25 nanogram, and for CSI we injected only 0.2 nanogram of the sample. So you can see here the sensitivity of both the molecule when we inject it in LC and then CESI. So when we calculate the concentration of the sample injected in both the technology and the sensitivity, so what we found here is in CSI, since uh, we are taking less number of sample, instead of that we are getting a very good resolution and the sensitivity is high. It is 500 fold high when we compare to LCMS. So the impurity profiles observed with both the methods are very different. If you see, we are getting many peaks in CSI, but in case of nano LC, you can also see the oxidized form of the peptide, which is not able to see in case of CSMI. So because this kind of information what we're getting, maybe because we have very less amount of injected sample and the flow rate is very small. So the ion separation is very less in case of CSI. So we will not able to differentiate these oxidized peptide in the CSI, but the same kind of information you can get in both the technology, and the sensitivity is very good in case of the CSI-MS. So in conclusion, the CSI-MS method based on the sheetless CE, which interface with the CE and the mass spec. And for this kind of application, we have dedicated different capillaries. So in this application, we use positive coated capillary. So we coat the capillary with polyethylamine chemicals, which give positive charge to the capillary, and then we can quantify the molecule by using the CSI technology. So the main advantage of this technology or this study was to analyze orthogonality and to check the orthogonality in the separation principle of CZD to RPHPLC for peptide analysis. And here we expected to detect a different impurity profile, and which was confirmed by this method. So none of the impurities found with CMS could be found with LCMS and vice versa. So here we conclude that the analysis of RPHPLC purified peptide is complementary by CMS by revealing a different impurity profile. But the sensitivity what we get here is 500-fold greater than the nano LC. So in SCIEX application demo lab, we are working on different peptides and hormones. These are the few molecule lists where we are using capillary electrophoresis technology for further characterization of these peptides. So you can see the name of the peptide and hormones. So the first one is insulin and insulin analogs like liraglutide, semaglutide. We are also working on ACTH, PTH, exenatide, glatiramer, somatropin, dulaglutide, and pramlintide. So these are the few of the examples where we are working, and whenever the methods are ready, so you can also see the tech notes and we will share the methods with you. So we have taken different references for this study, and you can find out the different references. These are the very good material to study, and you can also find the different protocols, challenges in these references. So that's it from my side. Thanks for listening. Thank you.